You said some fascinating research my way about antimicrobial peptides.
AMP.
AMPs, yeah.
Right.
And given, this rise in antibiotic resistance, it feels like we're always looking for new weapons.
Mm.
And this research suggests there might be a whole new arsenal
Yeah.
hiding in plain sight.
It really is groundbreaking stuff.
So
AMPs are these small proteins that are produced by microbes to kill other microbes.
It's microbes fighting microbes.
Exactly. It's like a microscopic war zone out there.
So we're harnessing the power of their weapons.
That's the idea. And what's really exciting is the scale of this discovery.
Okay.
We're talking potentially 1 million new antibiotic candidates.
1 million. That's almost impossible to fathom, like how...
I know, it's mind-boggling, isn't it?
Yeah.
And the key here is the way they did it.
Okay.
The methodology.
Yeah.
Researchers use machine learning to analyze a massive data set.
Yeah.
Over 63,000 metagenomes and almost 90,000 genomes.
Wow.
So they created this enormous catalog of AMPs. We're calling it AMP sphere.
AMP sphere. I like it.
Yeah, catchy, right.
Yeah.
And it contains a staggering 863,498 non-redundant peptides.
So just to put that in perspective,
Yeah.
most AMP databases just have a few thousand.
Exactly. This is a major leap forward.
So this is like discovering a whole new continent of potential antibiotics.
It really is.
And you're saying most of these AMPs are totally new to science.
Yeah, the estimate is only about 9% were previously known.
So where are all these AMPs? Like, where are they concentrated in the environment?
Well, they're found in a variety of environments.
Okay.
Soil,
Yeah.
water, plants,
Plants, too.
even the human gut.
Wow.
And interestingly, the top contributing genus was Prevotella,
Okay.
which is common in animal guts.
So are you suggesting that there's a rich source of AMPs
Potentially.
within host-associated microbes?
Seems like it.
That's fascinating.
It is, isn't it?
Yeah, so thinking about their role in the microbial world, how did these AMPs evolve in the first place?
Well, researchers have identified two main mechanisms. One is called truncation,
Truncation.
where mutations essentially chop off parts of longer genes,
Okay.
leaving behind a smaller functional AMP gene.
So it's like evolution found a shortcut.
Yeah, it's like evolution stumbled upon a way to create these powerful weapons.
Fascinating. And what's the other mechanism?
The second one is duplication.
Yeah.
Genes get duplicated, and then the copy mutates and evolves into an AMP gene.
Mm-hmm.
A good example of this is ribosomal proteins.
Which are involved in making other proteins.
Right, exactly.
So it seems evolution has been crafting these AMPs through both chance and adaptation.
Precisely, and what's really interesting is that a significant portion of these AMPs are entirely unique.
Oh, really?
Yeah, they don't resemble anything in the current databases.
That's amazing.
It really is.
It makes you wonder how those tactics vary.
Yeah.
Are these AMPs effective against a broad range of bacteria, or are they more specialized?
That's a great question, and it turns out most of them are quite specialized. Yeah. Often found only in specific habitats or even specific strains of bacteria.
So not a one-size-fits-all approach?
Nope, not at all.
Okay.
And this specialization actually ties into the concept of the pangenome.
Pangenome.
Yeah, the core genome is what all members of a species share,
Wow.
while the accessory genome varies between individuals.
So the accessory genome is where things get more personalized.
Exactly.
Okay.
And AMPs seem to fall primarily within that accessory genome.
That explains their specificity.
Right.
So, for instance,
Yeah.
a specific AMP called AMP 10.018194
Right.
was only found in certain strains of Mycoplasma pneumonia.
That's right.
So rather than being randomly distributed, these AMPs are strategically placed
It seems so.
within the microbial world.
Yeah.
It's almost like each strain has its own unique arsenal.
Yeah, like a personalized weapon system.
But does this specialization have any implications for how these bacteria interact?
Well, there's a fascinating connection that the researchers uncovered. The bacteria that produce more AMPs seem to be less transmissible from person to person.
That seems counterintuitive.
It does, doesn't it?
Why would bacteria that are better at fighting be less likely to spread?
It's a bit of a puzzle.
Yeah.
But one possibility is that AMPs might help bacteria fiercely defend their niche,
Okay.
making them less likely to venture out and spread to new hosts.
Interesting.
Yeah.
So it seems there's a trade-off between being a fierce warrior and a successful colonizer.
Exactly.
But all this talk about microbial warfare tactics begs the question, do these AMPs actually work? Can they really kill bacteria in a way that's useful to us?
That's the critical question, and the researchers didn't just stop at computational predictions.
Okay.
They actually synthesized 100 of these AMPs
Oh, wow.
to test their effectiveness in the lab.
And what did they find?
The results were impressive. Out of the 100 AMPs tested, 79 showed activity against at least one pathogen.
So a pretty good success rate?
Yeah, definitely.
Were any of them particularly potent?
Absolutely. Some were surprisingly potent, even rivaling known antibiotics in their effectiveness.
So could we have stumbled upon a weapon against superbugs?
It's certainly a possibility. They even found AMPs that were effective against vancomycin-resistant Enterococcus.
Which is a major concern in hospitals.
A huge concern.
This is really promising, but I do wonder about potential downsides. Did any of these AMPs also target beneficial bacteria,
Yeah.
like the ones in our gut?
That's an important consideration, and the researchers did find that some of the AMPs were active against good gut bacteria,
Okay.
which is somewhat unusual for AMPs.
So there's potential for collateral damage.
There is.
Something to keep in mind.
For sure.
But let's delve into how these AMPs actually work. What's their mechanism of action?
Well, the researchers found that most of these AMPs disrupt the bacterial membrane.
Okay.
Think of them like highly specialized detergents
Right.
that target specific components of the bacterial membrane, causing it to break down and the cell to die.
So it's a very targeted attack, which might explain why some of them can evade bacterial resistance mechanisms.
That's right.
But did they only test these AMPs in a lab setting, or did they actually see how they performed in a living organism?
They took it a step further and tested some of these AMPs in mice with skin infections.
So they moved from the petri dish to a more realistic model.
Exactly.
That's exciting. What were the results like?
The results were encouraging. Several AMPs proved effective in fighting the infection, significantly reducing the amount of bacteria present.
So these AMPs can actually work in a living organism.
They can.
That's a big step forward. Were there any standout performers in these mouse trials?
2 AMPs were particularly impressive: lecnosporin 1 and enterococin 1.
Okay.
They managed to reduce bacterial counts by a staggering 3 orders of magnitude.
Wow, that's significant. So we've gone from discovering this hidden world of AMPs to actually seeing them work in living creatures.
I know, it's incredible, isn't it?
It feels like we're witnessing a scientific breakthrough in action.
It truly is groundbreaking, and it highlights the immense power of machine learning for scientific discovery. It's like having a powerful new lens to explore the microbial world and unlock its potential.
That's a great way to put it, and this is just the beginning.
Right.
Who knows what other amazing AMPs are still out there in the AMP sphere,
Exactly.
waiting to be discovered.
There's a whole universe of possibilities out there.
It's exciting to think about.
It really is. It is a tantalizing concept, isn't it?
It is.
Imagine being able to analyze the specific bacteria causing an infection and then selecting the AMP.
Right.
That's most likely to be effective.
That level of precision would be revolutionary.
It would.
Instead of using broad-spectrum antibiotics
Right.
and hoping for the best,
Exactly.
we could be much more targeted in our approach.
Precisely.
But I imagine there are significant challenges in translating this research into personalized treatments. What are some of the hurdles we need to overcome?
Well, one of the biggest challenges is understanding the complex interactions within microbial communities.
Right.
It's not as simple as just finding an AMP that kills a particular bad bacteria.
Yeah.
We also need to consider how it might affect the balance of the entire microbial ecosystem.
That's like an ecological puzzle.
It is.
We need to make sure we're not disrupting the delicate balance of the microbiome
Right.
while trying to eliminate a specific threat.
Exactly. And then there's the issue of delivery.
Okay.
How do we get these AMPs to the site of infection in a way that's both effective and safe?
Those are all important considerations.
They are.
But even with those challenges, the potential benefits are enormous.
They are huge.
If we can harness the power of these AMPs, it could completely change how we approach treating infections.
It could revolutionize medicine.
And it's not just about treating existing infections.
Right.
What if we could use AMPs to prevent infections in the first place?
That's a fascinating idea.
How would that work?
Well, we could potentially use AMPs to boost the body's natural defenses against pathogens.
Yeah.
For example, we could develop AMP-based therapies that strengthen the microbiome's ability to fight off invaders.
So it's like giving our microbiome a power-up.
Exactly.
That's incredible.
That's a really exciting possibility.
It is. And what about the idea of engineering new AMPs? Could we design them with specific properties to target certain bacteria or even enhance their effectiveness?
It's an area of active research,
Good.
and it holds immense promise.
Yeah.
By tweaking the structure of these AMPs, we could potentially create even more potent weapons against pathogens.
Okay.
Imagine designing AMPs that target multiple bad bacteria at once,
Wow.
or those that are less likely to harm the good bacteria.
It sounds like we're talking about a whole new generation of antibiotics.
We are.
And given the rise of antibiotic resistance, this research couldn't be more timely?
It's a game changer.
It is.
AMPs offer a glimmer of hope in the fight against antibiotic resistance
Mhm.
because their mechanism of action makes them less susceptible to resistance development.
Right, because traditional antibiotics often target specific bacterial processes.
Right.
But AMPs tend to disrupt the bacterial membrane,
Exactly.
which is a more fundamental and
Mhm.
harder to evade target.
It's like trying to outsmart a bacteria that's constantly evolving ways to dodge our attacks.
Right.
AMPs offer a different strategy entirely.
But I do wonder about the potential ecological impact of introducing engineered AMPs into the environment.
Yeah.
Could there be unintended consequences?
That's a valid concern. Whenever we manipulate biological systems, there's always a possibility of unintended consequences.
Right.
It's crucial to proceed with caution and conduct thorough research to understand the potential risks and benefits.
Absolutely. We need to make sure we're not creating new problems while trying to solve existing ones.
Right.
This deep dive into AMPs has been eye-opening. It's made me realize just how much we still have to learn about the microbial world
There's so much we don't know.
and how much potential it holds for solving some of humanity's biggest challenges.
It's a vast and incredibly complex world, and we've only just begun to scratch the surface. This research is a reminder that even in the face of a growing crisis like antibiotic resistance, there's always hope for new discoveries and innovative solutions.
Well said. This conversation has left me feeling both excited and humbled. Excited about the potential of AMPs and humbled by the complexity of the microbial world. But as we wrap up our exploration of AMPs, I think it's important to step back and consider the broader implications of this research.
Okay.
We've been talking about the potential for new treatments, but what about the ethical considerations of manipulating microbial ecosystems? Where do we draw the line between harnessing the power of nature and potentially disrupting the delicate balance of life?
That's a crucial question, and it's one that we need to grapple with as we continue to explore the potential of AMPs. We're essentially talking about shaping microbial communities to our advantage, and that comes with a certain level of responsibility. We need to ensure that we're using this power wisely and ethically, considering the long-term consequences of our actions.
I agree. It's like we're stepping into a new era of scientific exploration.
Yeah.
And with that comes a need for thoughtful reflection and careful consideration of the ethical implications. But despite the challenges and complexities, I can't help but feel a sense of optimism about the future of fighting antibiotic resistance. The discovery of the AMP sphere opens up a whole new world of possibilities, and I'm eager to see where this research leads.
Me too. This is just the beginning of an exciting journey into the vast and largely unexplored world of AMPs, and as we continue to delve deeper into this fascinating realm, I have no doubt that we'll uncover even more groundbreaking discoveries that have the potential to transform medicine and beyond.
Yeah, it's a delicate balance, isn't it?
It is.
Pushing the boundaries of scientific discovery,
Yeah.
but also respecting the interconnectedness of life.
Absolutely.
But I think that's what makes this research so compelling. It forces us to confront these big questions.
Yeah.
And think deeply
For sure.
about
For sure.
the potential consequences.
It's a reminder that science isn't just about making discoveries. It's
Yeah.
also about understanding their implications and using them responsibly.
Right.
And when it comes to the microbial world, that responsibility is even greater
Because
we're dealing with a vast and intricate web of life
right.
that we're only just beginning to comprehend.
It's a humbling thought, but despite the challenges and the complexities, I can't help but feel a sense of awe at the sheer ingenuity of nature.
Yeah.
These AMPs, these tiny proteins with the power to kill bacteria,
It's amazing.
are a testament to the incredible creativity of evolution.
I completely agree. And the fact that we're only just now discovering the vastness of the AMP sphere speaks to how much more there is to learn about the microbial world.
It's like we've opened a door to a whole new realm of possibility.
Yeah.
I'm excited to see where this journey takes us.
Me too. It feels like we're on the cusp of a new era of scientific exploration, one that's driven by a deeper understanding of the interconnectedness of life.
And as we continue to delve deeper into this fascinating realm, I have no doubt that we'll uncover even more groundbreaking discoveries that have the potential to transform medicine and beyond. It's been an incredible journey exploring the world of AMPs, from their discovery to their potential to revolutionize medicine and even reshape our understanding of the microbial world. We've discussed their evolutionary origins, their specialized nature, and their effectiveness against even the toughest bacteria foes we've even touched upon, the ethical implications of manipulating microbial ecosystems. And while this deep dive has come to an end, the story of AMPs is just beginning. So to our listeners, I encourage you to keep exploring, keep questioning, and keep diving deep into the fascinating world around us. Who knows what other secrets are waiting to be uncovered in the vast and mysterious realm of the microbiome?